So everything here says it's 9. First thing you can do is put away your clickers, because I'm not sure what we would be asking clicker questions on other than more questions on the midterm. I think there were enough of those. Speaking of the midterm, I've got a couple of questions about where we stand grading-wise. Now I have a better idea that there are at least 2 ringers in this class. You know who you are. So that means that the scaling is actually not going to be to 86, which is what the high score would be. It's actually going to be to 80 or potentially lower for the 2 exams that you count together. Not having clicker questions today, but I did post updated clicker scores to D2L about 20 minutes ago. So those are there. There's some weirdness because some people seem to have gotten 4 points, even though there are only 3 questions. So don't consider yourself lucky with that. I will be going back and changing those. So are there questions about sort of grading, et cetera, as far as the midterm is concerned at this point? Otherwise, please feel free to email me. A lot of people have set up extra office hours. There are a couple of questions, however, on the midterm that I wanted to go over. The first one of those is the terminally redundant parts of a genome. And the question, slightly paraphrased, is if one end of a terminally redundant genome has genes for endonuclease, exonuclease, and DNA polymerase, what must be at the other end of the genome? And this has to do with a general definition of terminal redundancy. So each of these genomes here is terminally redundant. Here, you've got A at both ends. You also have B, but you certainly don't have C at both ends. Here, you've got B at this end. You have B at that end. Terminally redundant means whatever's at one end is going to be at the other end. It doesn't mean that all of the genes at one end are going to be at the other end. So that was what I was aiming at in that question. But I think about 20 people ended up getting it right, if that, and those probably guessed. So it was probably a bad question. So second one, and this I think is actually more important just in terms of thinking about how sequencing works. And so to say, for capillary Sanger sequencing, you need 8 different nucleotides in one reaction. And the 8 different nucleotides you need are each of the 4 DNTPs and 4 dideoxy NTPs. So if you're doing capillary sequencing, that is, you've taken all 4 of your normal reactions that you do for Sanger sequencing and put them into one reaction and won them on one gel equivalent, which is that capillary. And I think there was some confusion about what I meant by capillary in terms of capillary sequencing. That capillary is just a really small, usually plastic tube, but usually people think of them as glass. And then in that you have a gel which separates each of the nucleotides one base at a time. And so depending on what that terminal nucleotide is on their fragments that you're getting out of there, that's going to tell you what your sequence is. You need 4 DNTPs and 4 dideoxy NTPs. And then the last question I wanted to mention, again, these are the ones that most people ended up missing. Dual selection is critical for ES-cell knockouts because there's lots of non-homologous recombination, and that means you have to select for recombination in general, and that's that first part of selection, and then against non-homologous recombination. And so you need to do 2 separate sets of selection, and this is the slide that basically outlines that. If you're doing ES-cell transformations, you transform them with your favorite gene here, and this is basically what you're trying to do the knockout. So that's that gene right here you're trying to knock out. You've got an antibiotic resistance in that gene. You do transformation, so you first have to find the cells that picked up this DNA at all. And so then you have to select for your antibiotic resistance, in this case neomycin. Here, there are 2 possibilities. Either you have homologous recombination, which is what you want, or you have non-homologous recombination, which is what you don't want. Because here, this gene with its antibiotic resistance is in the genome that you get down here, but it's not knocked out the gene that you wanted to knock out. And the way that you control for that is now you select against these guys which have had this extra insertion in here. So you have to have 2 different selections. This is the dual selection that you need for that particular knockout. So there are questions on these questions. Now, hopefully that's a little bit more obvious. Okay, so I just noticed that the outline here is wrong because I didn't edit it. So this is the outline for today's lecture, which should be at least part of it. We'll talk about telomeres because I think telomeres are absolutely critical in terms of understanding what goes on at the ends of eukaryotic chromosomes. And we'll also talk about bacterial transcriptional regulation. And this is probably a good time to remind those of you who haven't left yet that I won't be here next week. So we'll have guest lecturers all next week. Either Dr. Bartlett, who many of you will have had for Principles of Biology. He'll be lecturing on Monday and Friday. Graduate student who you've seen here before, Eric Iverson, will be lecturing on Wednesday. And then Rahul Raghavan, who's a new member of our department, but is a real specialist in small RNAs, will be lecturing on small RNAs next Monday. So again, I'll be out this particular week, and if you're wondering why I'll be gone, talk to the instructor of cell biology next term. He can tell you all about it. They will be posting, hopefully, all the lectures, at least beforehand, lecture notes on D2L, and I'll be uploading at least my raw versions. Those may be changed. I'm not sure if they'll be recording the lectures and putting them up online. That's really up to them. Clicker questions, I've said if they want to do them, they may or may not. So, but at least the clicker questions we have. We will have clicker questions on Friday. Okay, other questions? You had one in the back? Yes, so I'm trying to get that to work. Whether it will or not, I'm not entirely sure. I know my graduate student is planning on doing it. The other ones I'm not sure about. Okay, so let's move on and talk about the end problem. And so this is the beginning of talking about telomeres. So basic problem with the ends of linear genomes has to do with the fact that DNA polymerases can only extend from primers. And those primers for DNA polymerases are always RNA. Well, those RNAs get degraded. And so what that means is if you have any end of any linear genome, chromosome, you name it, you have a problem, because to replicate this strand, you need to make a primer, and this primer is RNA, and that RNA is going to get degraded. Once this RNA is degraded, you can't make any more of this particular piece of DNA because there's no primer over here. There's nowhere to start with. And so this is why linear chromosomes are a big problem. Now, the way that we have dealt with these linear chromosomes, and pretty much most organisms with linear chromosomes, is making telomeres. And so telomeres are just the sequences that you have at the end of your linear genomes. And all that they are highly repeated sequences. And it depends on what organism you actually happen to be, what that repeated sequence is. So here in humans, it's TTA, GGG. I don't try and remember that sequence. But there are many, many copies of it, usually hundreds and sometimes even as many as thousands of copies. In other organisms, you have other sequences. So that's just that it doesn't seem to matter what that actual sequence is. It's just you have many copies of a sequence. Now, how do you have those sequences? Through the action of telomerase, which is one of my favorite enzymes. It's a reverse transcriptase. So it takes an RNA template and makes a DNA copy of it. Now, the really cool thing about telomerases is they actually have their own template that there's part of the telomerase enzyme. And that template, of course, is RNA. And so what that means is this is a ribonucleoprotein complex. So it's got a protein and an RNA. And that RNA is providing the sequence, which is what's making all these copies in the telomere. So that you can see over here in our telomerase RNA sequence, which is, again, the template for that reverse transcriptase. It has the complementary sequence in here for your telomere. So in this case, TTA GGG, again, opposite strand going the opposite direction, because always we're doing 5' to 3' opposite directions. We have here for the human telomerase, starting here, TTA GGG. So cartoon form, what does this look like? We've got our green part here, which is our telomerase enzyme, which again is a DNA polymerase. So it will extend from already present 3' OHs, but just copy on the template which it has. And again, that template is a RNA. So, okay, but wait a minute. It's extending on 3' ends. We need to deal with the 5' end, right? That's the one that keeps getting shorter and shorter because we've chewed off the template. So what happens in this case is you have multiple copies that get added on your 3' end, because it's the 3' OH. So now here we have the telomerase, which is just extending and making multiple copies now. And this is 4G's TT, 4G TT. Again, it doesn't matter. You're just making multiple copies here. Now you end up with many, many copies now on your 3' end. Now this is enough of a template that you can put your normal DNA polymerase on the other strand. And so that can fill in. And that's the normal DNA primase, which is an RNA polymerase. We'll put in a little bitty piece of RNA. And then it'll get extended by the normal DNA polymerization machinery. So all that the telomerase is doing is it's making more template. It's not adding on to the end, which is getting chewed off. It's making more template so that you can fill in that end. And basically all it's doing is it's pushing the problem further and further out. And so you're always going to have a little piece at the end which is a single strand. Now this, however, turns out to be a blessing in disguise because this particular sequence now can come back in base pair with that opposite strand that you've made. And this will generate something called a T-loop or a telomere loop. So this extra 3' end, which you're always going to have because that cellular DNA polymerase can't always replicate out to the end because it's losing its primer, that can now come back and base pair and give you this loop structure which protects the ends of your DNA. And we talked about the exonucleases. So these are the nucleases that chew in at the ends of pieces of DNA before. There are lots and lots of them in the cell, and protecting ends is a really, really critical thing to do. And so this is the way that this happens. This T-loop is only one part of a structure of a whole bunch of proteins that sit down at telomeres. Another thing that happens at telomeres is that you end up with heterochromatin, which forms at the end here. And so we talked about heterochromatin before. That heterochromatin, the constitutive heterochromatin, i.e. stuff that's compacted all the time, there are two places that you find that. One's at the telomere, and the other one's at the centromere. So questions about telomeres before we switch gears completely and talk about bacterial transcription. Yeah?
So the exonuclease is part of this, or it protects from the exonuclease?
Yeah, so the question here is an exonuclease part of this, or is this protection from the exonuclease? It's protection from the exonuclease. So there are no exonucleases that are part of that telomerase complex, or telomere complex, I should say. Yeah?
So if it's binding there at the end, is it interrupting the other binding area of the DNA? Are those base pairs bond down one strand or the other, or is it bonded usually both?
Okay, so I guess the question is, as far as this T-loop is concerned, right here, what are these guys base paired to? We're always going to have that 3' extension again, because you've deleted the RNA part of the primer, which is there. But that sequence, because it's been copied from your telomere template, it's always going to have multiple copies of exactly the same sequence. And so that's going to be able to base pair with some of the sequences which were much earlier on. So say this will be repeat number 100. It can come back and base pair with the template for repeat number 1. Yeah? Yeah, no, so this is in fact not forming a holiday junction. The question, so is it forming a holiday junction? No, it's just strand invasion. So it's sort of like the first step of what you'd have for homologous recombination. But to have a holiday junction, you have to have two double strands which are attached to each other. And here's just a single strand which has come in there. And that's part of what all those extra proteins are there doing, is stopping that process. And I'm not going to throw them at you. It's way too complicated. We're complicated enough as far as this course is concerned. OK, so let's move on and talk about transcription. This is basically, this slide anyway is review. All that transcription is making RNA from a DNA template. So templated polymerization, RNA polymerases are just like DNA polymerases. The chemistry is practically identical. The only thing is you have a ribose instead of a deoxyribose as part of your nucleotide triphosphates. You have uracil instead of thymine. But the real key here is you don't need a primer. So unlike all of the DNA polymerases, and that includes telomerase and all the reverse transcriptases, these guys can start from scratch. They can start pretty much anywhere in the genome. And this is really great because you can start anywhere, but it also means you have to have ways of figuring out exactly where to start. And so it adds an extra layer of complexity in terms of thinking about these things. Where do you start? Of course, these are promoter sequences. So this is a classic promoter here, just what I mean. I'll be talking a lot about upstream and downstream as we move along through this lecture and the lecture on Friday. It's all relative to the start site of transcription, and that start site of transcription is the +1 nucleotide. And again, this has a triphosphate at the 5' end. So again, starting from scratch. Once you've started here, this is your +1. You'll move, and we'll see the whole polymerase machinery will move along and polymerize using this template strand as a template. So going from 5' to 3', it's going to be the opposite orientation. And then making your RNA copy of what's also known as the coding strand or the sense strand. So copying, template. Again, pretty straightforward. Downstream is going to be the direction that you're transcribing. Upstream is going to be away from that direction. What does this job? This should also be called, I can get rid of this, the DNA-dependent RNA polymerase. We mentioned really briefly some of the RNA-dependent RNA polymerases. But this is a DNA-dependent RNA polymerase because it uses a DNA template and then makes an RNA copy from that. This is an overview of what that looks like. You have to pull the two DNA strands apart in order to be able to use one of those as a template. This is a temporary separation of the strands because they come back together after you come through the RNA polymerase. And then the RNA is made and it exits through a small hole in the side of the RNA polymerase. The nucleotide triphosphates come in, get incorporated here right in the middle of the protein. We can actually visualize this happening here. This is a DNA along the middle here, and this is now being transcribed. The transcription is happening with the RNA polymerase, which are these darker blobs along this line here in the middle. And then these blobs at the end are actually modifying that RNA as it's being made. And we'll talk much more about modification later on, and probably more next week when Dr. Bartlett and Eric Iverson are speaking. So if you look at this RNA polymerase, it goes through basically exactly the same steps that we've already talked about for replication and translation. Starts, moves along, and terminates. And this is, again, a whole cycle. And again, we've talked about this quite a bit already. So first thing that happens is you have to have your RNA polymerase, and that's this blue blob here, come together with the right place on the DNA, and that right place is the promoter sequence. How do bacterial RNA polymerases recognize this? It's all through this sigma factor. And so the sigma factor is just how named alpha, beta, gamma, it turns out there wasn't, and sigma, going through the Greek alphabet. So this sigma factor is absolutely required together with the RNA polymerase to bind to promoter sequences. And it's really that sigma factor which gives you the specificity. It tells the RNA polymerase where to bind. Once it's bound, then you have the first really committed step in getting transcription to take place, which is pulling the two strands apart. This should sound really familiar as far as replication is concerned. That's that committed step, is getting those two strands apart. But in the case of transcription, unlike when you have for replication, that helicase activity is due to the polymerase itself, at least for bacteria. We'll see it's slightly different in eukaryotes a little bit later on. So once you have these two strands that come apart, now the RNA polymerase can start making nucleotides. And this is where you really have one of the biggest differences between transcription, replication, and translation. RNA polymerases very often go through this step called abortive initiation. So they'll make a couple of nucleotides, and then they'll stop, throw out that set of nucleotides, and start again. And the reason for that is that these RNA polymerases, unlike your DNA polymerases, at least by themselves, are not processive. Or I'd say are processive. The DNA polymerases are non-processive. So the RNA polymerase is highly processive. So once that RNA polymerase gets on and starts to elongate, it's going to go all the way to the end of whatever gene it is that it's going to be transcribing. And that's a pretty energetically demanding process to go through. So you really want to be extremely sure that you want to make that piece of RNA. And so it seems that this abortive initiation step is really a way to be absolutely sure that you want to transcribe that particular piece of DNA. Because as soon as you move away from abortive initiation, this elongation complex is highly processive. It's going to crank along your DNA, making RNA, until it gets to the place that it needs to stop, which is the termination site. And we'll talk about these terminators a little bit later on. The sort of classic one is this hairpin loop structure, which basically pops the RNA out of the RNA polymerase. And we'll talk about two different ways to do that as we move along here. Transcription is directional. What do I mean by directional? It just depends on where your promoter is and what's upstream and downstream. The template is always double-stranded. And so depending on which strand you decide to use as a template, use the top strand or the bottom strand, upstream is going to be one way versus another way. And it turns out that this happens all the time. You've got many cases where you have promoters that can be on one strand in one part of the genome, promoters that are going to lead to transcription on the other strand, right next to them in different parts of the genome. Very often promoters are right on top of each other. So one going on one strand, the other going on the opposite strand. And we'll talk about why that is a little bit later on in terms of getting, mostly it's about regulation. So if you've got two promoters in one place, you can regulate both of those at the same time with the same proteins. So it's a little bit more efficient that way. If you look at the elongating polymerase, and this is one of the big breakthroughs in the last couple of years, we now have high resolution, basically atomic resolution structures for the RNA polymerase in most of the steps of this transcription process, the initiation process, the elongation process, etc. And probably most important from this elongating polymerase process here is that this protein just really clamps down around the DNA strand. And so it's kind of like the sliding clamp that you have in replication. You made a loop around your DNA. So this, once it's on, it's staying on. And so it's really a highly processive, and so we know exactly what the molecular basis is for this really high processivity. And we also know, again, that you've got holes that the RNA transcript goes through, et cetera. And so this particular structure, and Dr. Bartlett may talk a little bit more about this when he gives his lectures, this is something that is a really active area of research, and in fact the area of research that his lab is working on. So if we take that structure and we take it apart, I already told you a little bit about the sigma factor that we have down here at the bottom. But these RNA polymerases are made up of multiple different proteins. This is a really nice example of quaternary protein structure. So multiple different polypeptides that have all come together, and you need all of them to actually make this structure. You have, in the case of the E. coli RNA polymerase, 2 alpha subunits, a beta, one that looks a lot like beta, which is called beta prime. Some people talk about this omega subunit. The omega subunit is actually just important for getting that really complex structure together. Turns out once you've put your structure together, you don't need that anymore. But the business end of your polymerase are really in these beta and beta prime subunits. That's where you have the polymerase activity. Sigma, we talked about, that's your specificity factor. And as we'll see a bit later on, alpha is really mostly important for regulation of this polymerase and getting the polymerase to the right place. So let's talk a little bit more about some of these sigma factors. Again, I said they're the specificity factors. They're what allow the polymerase to bind to the appropriate sequence. Well, what is that appropriate sequence? In the case of the sigma 70 subunit, now sigma 70 is the most common of the sigma factors used in E. coli. It's also called the housekeeping sigma factor. It's what is involved in most of the genes in terms of transcription in E. coli. How do we know what sigma 70 binds to? Well, basically what people have done is they've taken lots and lots of sequences of promoters and then just lined them up with each other and done multiple sequence alignments. And then what they find is that at about 70% of the time, relative to your transcriptional start sites, this is now all upstream, at minus 35, you have mostly TTG ACA, but not always. Most of the time you have that. And then about 10 nucleotides upstream of your transcriptional start site, you mostly have TAT-AAT. Now again, this is just mostly. So you've got multiple sequences you've lined up with each other. In this case, it's hundreds of promoters that you have in the E. coli genome. And you can see just this distribution. And what you can see is now people have taken these promoters and then put them in front of reporter genes, as we talked about before. If you take one of these promoters that is close to that sequence that you find in most of these promoters, then turns out you get lots of transcription of that reporter gene. If you take a sequence which is not as similar to this sequence, it ends up being a relatively weak promoter. So you get relatively small amounts of transcription of that gene that is being regulated by that particular promoter. So this sequence here, down at the bottom, TTG-ACA and TAT-AAT, again, don't remember those sequences. Don't bother. I can't remember them. Can't expect you to. But you've got at minus 35 and minus 10, relative to the promoter, you have these conserved sequences that are going to be important for binding of sigma 70. Another thing that you find when you look at these promoter sequences is almost all of them are separated by 17 nucleotides. 17 nucleotides, that's a really funky number. But it's actually quite close to 20 nucleotides. And what is 20 nucleotides? What would that be? 2 turns of your DNA helix, exactly. And so what that means is basically the sigma factor is sitting down on one side of the DNA, even though it's binding to these sequences which are quite a long way apart. So we've got these multiple spacings that you have here. And it turns out that as well, if you now take minus 10 and minus 35 sequences that are 17 bases apart, they work better than sequences which are either 18 bases or 16 bases apart, which is going to be, again, each time you move, you're going to be moving around your DNA helix. And so 17 bases, those 2 sequences are right on the opposite sides of each other. Or I say same side as each other, I should say. So I mentioned sigma 70 is the main sigma factor that you find in E. coli. You should have immediately thought, well, okay, that means there must be some other ones. And yes, there are. This is just a list of some of these other sigma factors that you find not only in E. coli, but also in other organisms as well. And basically, having a different sigma factor means that now the RNA polymerase, which is the same, has now a different specificity. It's going to bind to a different promoter. And it turns out that you need to use these different promoters under different conditions. And just one of the examples here is this one, sigma 32, probably the best studied of these, which is a protein which is involved in heat shock. And what happens when you have unfolded proteins. So you remember unfolded proteins, what do you need to make to deal with that? Dealing with unfolded proteins. How do you deal with unfolded proteins? Chaperones, exactly. So what kinds of genes would you expect to be regulated by a promoter controlled by sigma 32? Heat shock chaperone genes. So the chaperone genes are going to be regulated by promoters that have this sigma 32 sequence that gets the polymerase to bind there. And again, the sequences here are not critical, but what's important is that the minus 35 region has a very different sequence here for sigma 32 than it does for sigma 70. And the same thing is true for the minus 10 region. So same polymerase, different sigma factors can bind to these different promoters under different conditions. And this is just shown as a cartoon form here. Here we have the RNA polymerase, much more blobby at this point, together with the sigma factor. This sigma factor allows both of these guys together to bind to the DNA and have helicase activity. Once you have the helicase activity, now you have the RNA polymerase, which can extend. You actually lose the sigma factor. The sigma factor is no longer bound to the promoter at this point once you move into this elongating form. And that elongation step actually is a change in the structure of the RNA polymerase. And we now know this again from some of these high-resolution structures. There's a pretty massive change in the structure of the polymerase. And one of the main things is that arm reaches down and closes the loop around the DNA that's coming into your RNA polymerase. And in doing that, you lose the binding of the sigma factor. The other important thing here is that the sigma factor by itself doesn't bind to DNA. It only binds together with the RNA polymerase. And just real quick terminology here, I'll try not to use this too much, but this is known as the RNA polymerase without the sigma factor is called the core polymerase, and with the sigma factor is called the holoenzyme. But yeah, I'll try not to use that terminology, but sometimes I may slip up and I may use some of these things. So now let's talk about the sort of three different aspects of, main aspects anyway, of transcriptional regulation: initiation, elongation, and termination. Initiation, as I mentioned before, as well as that abortive initiation, initiation is really where you want to have most of the regulation, because that's where you decide what gene is going to be turned on, when it's going to be turned on, and the cell has made the energy commitment to making that copy of that particular gene. So the first thing that happens is you have the, I'm going to try not to use it again, holoenzyme. So the RNA polymerase plus sigma factor binds to the DNA, and that's what's called a closed complex. And the only reason it's called a closed complex is because the two DNA strands are still connected to each other. The next step that happens is you have open complex formation, and open complex is just getting those two strands apart. So helicase activity of the RNA polymerase, and then you have abortive initiation that takes place, which I mentioned already, and then you finally move from that, that's getting started, are we going to do this, are we not going to do this, to actually moving beyond the promoter. You lose the sigma factor, you stop doing this abortive initiation, and then you're an elongating, highly processive polymerase. Now, you might wonder why we've got all these extra steps that are involved here in this whole process. Basically, each of these individual steps is a place that you can have regulation. You can have, okay, we'll regulate just binding of the polymerase to the promoter. We'll regulate how often you get that open complex that forms. We'll regulate how often you do abortive initiation, and you can regulate how you get promoter escape. And so this gives you really 4 different possibilities in terms of how you can determine how much of a particular RNA you're going to get. So most of the regulation, again, happens at this very early step, and we'll talk more about that, we being Dr. Bartlett, next week. During elongation, there is a small amount of regulation. Mostly, the RNA polymerase just chugs along the DNA template and spits out the RNA at the end. But there is a sort of equivalent to proofreading that happens during transcription, and that is when the RNA polymerase, as it's transcribing along the DNA, will get to a certain spot. This can either be a particular sequence, it could be actually some regulatory proteins that are associated with the DNA or the RNA, and pause and just basically stop. And in many cases, what that polymerase will do is actually back up. But instead of what happens with backing up and the replication process, where you've got proofreading, which just chops off a single nucleotide, so it's an exonuclease activity, the RNA polymerase actually has an endonuclease activity where it'll cut back, depends on the organism, but 10 to 12 nucleotides away from the end and chop off this RNA and then restart from here. And this is, again, just seems to be a regulatory process. And again, it'll depend on the DNA sequences and the RNA sequences as you're making those particular RNAs. So again, usually elongation goes along perfectly happily, but occasionally you'll have these pause and regulatory sequences, which can either be proofreading, or in some cases, you can actually get termination that will happen at these pause sites as well. But the normal termination is going to happen, of course, after you get to the end of your gene. And there are really, at least in bacteria, two ways that this happens. The so-called intrinsic terminators, which are probably what you've seen before. Hopefully you've talked about these or remember some of them from Principles of Biology. It's basically what's called a stem loop structure. So the stem is where you have a bunch of potentially base paired, and this is now in your RNA sequence, so double-stranded RNA from the 5' end going to the 3' end. These guys can all base pair with each other, and then usually there's a whole run of uracil residues that you have at the 3' end of this stem loop structure. Now, uracil, why uracil? So uracil is going to base pair with what? Adenine. How many hydrogen bonds does that make? 2. So it's less hydrogen bonds. So basically the bonding now between your RNA and the template is going to be weaker here. And this structure, if you remember that hole that the RNA is going through in the RNA polymerase, well, if you form this double-stranded structure, that can't fit through that hole anymore. And so basically, if you make these double strands, you're going to start to try and pull the RNA off of that DNA template. And so this process, forming that double-stranded RNA, and that you've got this stretch of U's here, will pull the RNA out of the RNA polymerase. Once you have no RNA there anymore, those two strands come back together, the polymerase falls off of your DNA, and you can start again. So that's one kind of termination that you have in bacteria. The other kind is so-called protein-dependent or factor-dependent termination, and this happens through a protein called Rho. I guess all the E. coli biologists were really into Greek at the time, so they'll give them Greek letters for their names. This is a ring-like structure which has a small hole in it. That hole is just the right size for single-stranded RNA. So it can bind to the messenger RNA as that messenger RNA is being made. How does it know where to bind to? Well, there's something called a Rho utilization site, and this is the so-called RUT site, Rho utilization, RUT, that's in the RNA, and once this Rho binds to the RUT site, then it will move along the RNA, and moving along the RNA in the same direction as the RNA polymerase, and basically as soon as that Rho factor catches up with the RNA polymerase, then, just like you have with that double-stranded structure, which is going to pop the RNA off, it basically pulls the RNA out of the RNA polymerase. And Rho, not surprising, has ATPase activity, and that's that ATPase activity which is used to translocate along the RNA, and once it bangs up against the RNA polymerase, pull that RNA out of the structure. So that's initiation, closed complex, open complex, abortive initiation, escape, elongation with pausing and backtracking, and then two kinds of termination that we have. Questions on initiation, elongation, termination. So initiation is where we're getting most of the regulation. So for the rest of today's lecture, we're going to talk about a couple of different examples of regulation. And the first one we'll talk about is the trip operon. So trip for tryptophan. Each of these enzymes which are encoded in one RNA encode enzymes that are important for making tryptophan. Tryptophan, some of you may remember, is one of the more complicated, certainly structurally, amino acids. It's got a couple of ring structures, etc. So it turns out it's actually really pretty expensive to make. You need multiple different enzymes, 5 enzymes in the case of E. coli to make tryptophan. Making an enzyme means you've got to make the RNA, you've got to make all the proteins. Again, this is a really energy-intensive process just to make one amino acid. Happen to have that one amino acid around, why bother making it? So this is a really nice example of why you want to regulate getting that particular, tryptophan in this case. And the way you can do it is by regulating all of these enzymes at the same time. And the way that works is through this so-called operator sequence. And the operator sequence comes from this term Operon that Jacob and Monod came up with. And we'll talk much more about Jacob and Monod a little bit later on. So here, you'll notice that there's a promoter sequence and an operator sequence which overlap with each other. So pretty simple process. How do you get regulation of all of these genes? You basically stop binding to the promoter. What does this? It's the trip repressor, the tryptophan repressor. An inactive repressor can't bind to DNA, so the RNA polymerase will bind here. You'll make all of these genes. On the other hand, if you have an active repressor in the presence of tryptophan, it binds to the operator sequence, and that operator is right between the -10 and the -35. You can't get binding of the RNA polymerase to this particular promoter. So it's a pretty straightforward process. In the presence of tryptophan, which means you don't need all those enzymes, you have the active trip repressor, binds to the operator, stops any kind of transcription from taking place. We know in really nice detail exactly how this works. The trip repressor is a dimer, so it's got 2 individual peptides that come together, again, through quaternary structure. Each of those binds tryptophan. When they binds to tryptophan through an allosteric process, so binding one part of the molecule changes the structure in another part of the molecule, now you get the DNA binding parts of the molecule in exactly the right position to bind to DNA, surprise, surprise, 10 base pairs apart. So that's what's happening in the case of the trip repressor. Now it turns out that that's only one level of regulation that happens at the trip repressor. You think that's all you really need. But it turns out that there are 2 different kinds of regulation here. And that second kind of regulation is called attenuation. And attenuation is a very specific kind of regulation that happens in bacteria and probably in archaea as well. And this has to do with the fact that when you have transcription in bacteria or archaea, you have translation that happens at exactly the same time because there's no separation. There's no nucleus that you have in bacteria or archaea. So as you're making an RNA, that gets bound to by your ribosome, and you have transcription and translation happening at the same time. So you're making an RNA and translating at the same time. In eukaryotes, of course, it's a little bit different. So what you see here is with your promoter sequence, there's our promoter sequence here, our minus 10 and minus 35 sequences. Then there's actually quite a long piece of RNA which can be made here before you actually get to these genes that are important for tryptophan biosynthesis. And there's a small peptide which is made here that starts, as usual, with methionine. And there's a ribosome binding site right next to this. And then right in the middle of it, there are a couple of tryptophan codons. And what this means is, you get to these tryptophan codons, this piece actually will get translated, because again, the ribosome binding site, getting to these two tryptophan codons. Now, why is this important? Well, because it turns out that this particular RNA actually forms a very specific structure. And that specific structure looks like this, and this circle should be showing you something. What does this look like? Something we just talked about. Muttering, so it should be looking a lot like a transcriptional terminator sequence. It's got a stem loop structure with a nice stretch of uracils right next to this. So if this structure is formed by the messenger RNA, then you're going to terminate your transcription before you make all of these downstream genes that are important for tryptophan biosynthesis. Now, how do you get this secondary structure? Well, this secondary structure happens when you have enough tryptophan around that you can translate this particular piece of your RNA. If you can translate this piece of RNA, then the ribosome will have moved through these tryptophan codons and then gotten to this stop codon, which will give you this particular structure. If, on the other hand, you have very small amounts of tryptophan in the cell, you're not going to have very many tryptophans bound to tryptophenyl tRNAs. So the ribosome is going to get to this sequence right here with these two trips right next to each other, and it's going to get stuck because there's not enough tRNAs to bind there in order to get that ribosome to keep moving. So if the ribosome is bound right here, these guys are in the A site or the P site of the ribosome, then this guy can't form this secondary structure. So there's a different secondary structure that forms here. This is not a nice stem loop structure with a stretch of U's next to it. So now transcription continues. So let's just look at this in a cartoon form right here. As you're getting translation, if translation happens normally, you've got plenty of tryptophenyl tRNA around, then you move past these tryptophan codons and you extend, you get to here. We make our transcriptional terminator. The RNA polymerase falls off. If, on the other hand, the RNA polymerase is stuck right here because there's not enough tryptophenyl tRNA around, then you don't have this terminator sequence that is made, and the RNA polymerase continues and makes all of your tryptophenyl tRNAs. So two different kinds of regulation. First is a standard repressor. The second one is this attenuation process. And it's not just for the tryptophan Operon. It turns out it happens in many different cases, that you have transcriptional termination that's coupled to some kind of translation which is happening at the same time. So we already talked about this first case now where we have repressor binding. And again, repressor binding here blocks your RNA polymerase from binding. You can have two different kinds of repressors. You can either have a repressor like you have in the trip operon, when you've got binding of a ligand, which causes DNA binding. Or you can also have cases where binding of your ligand causes the regulator protein to actually fall off of the DNA. And we'll see this for the lac operon in just a couple of minutes. But at the same time, you also have what's called positive regulation. And it's basically just the opposite of looking at negative regulation. Instead of binding by a DNA binding protein, which is blocking the RNA polymerase from interacting, now you have a protein that helps the RNA polymerase to bind. So this is the process here of positive regulation. Now you have an activator protein bound right next to the promoter, and that helps the RNA polymerase to bind, mostly through the alpha subunits we talked about before in that multi-subunit protein complex. And again, you can have either a ligand that binds here, which causes the activator to not be bound, or binding of a ligand that helps the binding to take place. And we'll see that in just a second here. Curiously enough, Jacob Monod, who came up with that idea of the Operon model, they were not convinced, particularly Monod, François Monod, who apparently one of my professors in graduate school gave one of his very first talks at a scientific meeting where he actually talked about this kind of positive regulation. And François Monod was sitting in the front row, who had just gotten a Nobel Prize. And he presented his talk, and at the end of the talk, this graduate student, actually postdoc at the time, said, well, I think this is some evidence for positive regulation. And apparently, François Monod, again, sitting at the, Jacob Monod, excuse me, sitting in the front row, said, there's no such thing as positive regulation, and stormed out of the room. Turns out that this guy was right. There was positive regulation. That's why we're talking about it now. But just goes to show if you have a Nobel Prize, you may or may not always be right about these things. So let's talk about a nice example of how you can get positive and negative regulation. This is, in fact, how a lot of this positive and negative regulation was originally discovered through the study of bacteriophage Lambda. So we skipped Lambda when we talked about viruses before, but now we get to talk about it again. So one of the really fascinating things about Lambda, one of your more fascinating things, take virology next term, is that Lambda can replicate in two different ways. Either, if you look at this outside circle here, through a lytic process, which is where the virus binds to the outside of the cell, injects its DNA, that DNA makes many copies of the proteins, and then it lyses the cell, and you just go around this process again. Or, and this is the neat thing about Lambda, is it can go into this lysogenic cycle where the viral genome gets integrated into the host genome, and it just hangs out inside the cell, inside the host genome. And so it's really this decision right here, when infection takes place, whether to undergo this lysogenic cycle or this lytic cycle. And this is what's called the genetic switch. And it turns out it's completely regulated at the level of transcription. And we know about this regulation at the level of transcription here through the Lambda regulatory region. All the details on here are not critical, at least not this term. Next term they will be. But here, what's important are there are two main promoters. There's this PR promoter and this PL promoter. And it's just for right and left. And it's also a nice example of promoters going in opposite direction. So different strands giving you different proteins right next to each other. Now, why is this so useful? Well, it's because the operator for the rightward promoter and operator for the leftward promoter, are basically right on top of each other. Now, if you get expression from this rightward promoter, you end up getting your lysogenic development, and from the leftward, you end up getting lytic development. So it's this decision whether you go one way or the other way which is most important. So this nice, simple mechanism right here, basically the important thing here is this protein right here. The Lambda C1 protein, also known as the Lambda repressor. And again, we'll talk about all these other things next term when we talk about regulation in Lambda bacteriophage. Lots of this work was done by Ira Herskowitz, who you can probably tell was in San Francisco in the 60s. So what is this Lambda C1 protein? It's the so-called Lambda repressor, but repressor, as you can see here, is in quotation marks. And that's because the Lambda repressor can actually act as both an activator and a repressor. And all that depends on is exactly where the Lambda repressor has bound to DNA relative to where the promoter sequence is. So if we look here, let's look at the bottom one here. If the Lambda repressor overlaps the promoter, and that's what these bars down here are supposed to show, overlaps the promoter, it acts just like a normal repressor. And so that repressor will, turns out, repress all of the lytic genes, so expression of all the lytic genes which are going to lead to lysis. So this is going to lead to a lysogenic process. But it turns out that where the Lambda repressor binds, and this can actually be exactly the same binding site, now is right next to a promoter. So it now stimulates binding of the RNA polymerase. And it turns out this is just one base pair difference, where the Lambda repressor is bound, whether it's acting as a repressor, acting as an activator. And so again, this is a lot of how we know about what's going on in terms of transcriptional regulation comes from just this study of the Lambda bacteriophage and whether it's going to undergo its lytic cycle or whether it's going to undergo a lysogenic cycle. So last example I wanted to talk about is the lac operon. And this is, in fact, where Jacob and Monod did all of their studies and where they came up with the whole idea for the Operon model in the first place and determined that it was just negative regulation which was happening, just having repressor binding. Of course, later people showed that there was activator binding there too, which was a little embarrassing. But yeah, they're French, and so they don't care about these things. So I've lived in France for many years. I've got great French friends, so don't take that too personally. So this is the lac operon, and again, it's got 3 genes in it, a lot like the trip operon, but instead of the trip operon, which is important for making a particular amino acid, in the case of tryptophan, the lac operon actually encodes proteins that are important for breaking down lactose as an energy source. So here we've got the lac-Z protein, which is the beta-galactosidase that breaks down the actual lactose into glucose, which is what the cell needs, and then lac-Y and lac-A, which are for getting that particular sugar into the cell, and then an operator sequence right here. Considerably further away is this protein called lac-I, which is the lac repressor. So if you look at the lac operator, here is the transcription start site, again, +1. What's interesting is that in this case, the lac repressor actually binds downstream and a little bit upstream of the start site for transcription. So it's not sort of binding between -10 and -35 as you have with TRIP. It's actually binding right around the start site of transcription. But the important thing to look at here is that the binding of the lac repressor is binding to these inverted repeat sequences. So if you look here, AATTGT on one strand, on the opposite strand, AATTGT. Same thing here on the outside, TGT-GTG, TGT-GTG. And so the lac repressor is binding to these inverted repeat sequences. And you remember what I said about the trip repressor? It's a dimeric structure. And so these dimers are very often going to be binding to, and Dr. Bartlett will talk about this much more later, they're going to bind to what are called half sites. And so each of these are inverted repeat sequences relative to each other. But as far as the major groove is concerned, they're going to be identical in terms of hydrogen bond donors and acceptors. And so you have 2 dimers that are going to bind in opposite orientation to each other. They're going to be binding to inverted repeat sequences. And again, he'll talk much more about this a little bit later on. But they're inverted repeat sequences that the lac operator binds to. So if you look at regulation of the lac operator, in the absence of glucose and the presence of lactose, you get optimal expression of these genes. Now, why the absence of glucose and the presence of lactose? Well, okay, presence of lactose is pretty obvious. If you don't have any lactose around, why bother making any of the genes that you need to break it down? But what about the absence of glucose? Well, it turns out that if you've got glucose around, why do you need to bother going from lactose and making glucose out of it if you've already got glucose? And so basically what you need here is for the presence of lactose, and so presence of lactose binds to the lac repressor, and that binding to the lac repressor causes a structural change, but that structural change, instead of now binding when it's bound to ligand, is now falling off in the presence of ligand. So now your repressor is not bound. But you also need this activator protein, which will bind and stimulate the expression of this particular promoter. Now, that activator protein is called either CAP or CRP. Depends on what research group you happen to be working with. So it's either the cyclic AMP binding protein or the catabolite regulator protein. It binds to cyclic AMP. Cyclic AMP is made when cells are under starvation conditions, i.e. no glucose. So if you have no glucose, you make cyclic AMP. Cyclic AMP binds to this protein. You can see the little gray balls right here. Once it's bound, then this CAP or CRP binds to DNA. It binds to DNA, and again in this inverted repeat sequence, and you have transcriptional activation because this protein, this part of the protein right here, now interacts with the alpha subunit of the RNA polymerase and helps it bind to the DNA. So let's look at this process again. Here's the lag operator up here together with the CAP or CRP binding site. The operator is again slightly downstream of the promoter. In the absence of lactose, the lac repressor is going to bind to DNA. So no lactose around, you don't need to make any of these genes to break it down. That's going to stop any expression. In the presence of lactose, this lac repressor then is no longer binding to the DNA. And in the presence of glucose, you get a small amount of transcription that takes place here because the RNA polymerase can bind. It just doesn't bind very well unless you have CAP, CRP bound to cyclic AMP, which will stimulate the binding of this RNA polymerase and give you lots and lots and lots of transcription. So this is great, but there's also a potential issue if you have no glucose around and also no lactose, that you really want to make sure that this whole thing is completely shut down. You'll notice that there's a couple of extra tails hanging out on the lac repressor over here. It turns out that there's another operator site that the lac repressor can bind to that is both 400 base pairs downstream, plus 412, and another one which is 82 base pairs upstream. And so in the absence of lactose, what this protein does, the lac repressor, actually is going to compact the DNA and actually make a DNA structure which completely blocks access by any of your RNA polymerases. So it's not just the direct blocking of formation of your closed complex. It's also closing down the DNA to really shut down any kind of activation which could be taking place there. So that's the case for the lag operon. I'll talk about activation at a distance and eukaryotic gene regulation on Friday.